Hello everyone, welcome back to Zoology with Rituparna. In today's lecture, we will learn the basis of animal classification and some peculiar characteristic difference between chordates and non-chordates. So before proceeding, I want to share something with you that my academic background is from CHSE board. So I am comfortable both in English and Odia. If many of you would like to explanation in Odia as well, please let me know in the comments. Based on your feedback, I will be happy to make a bilingual videos so that learning becomes easier for everyone. So let's start today's class with a beautiful query, that is, have you ever think why a spongy, a butterfly, an earthworm, a fish and a human all are belong to kingdom Animalia, yet they are placed in different phyla? See, scientists classify animals based on several important characteristics such as level of organization, body symmetry, germ layers, body cavity means coelom, segmentation and notochord. So these characteristics helps us to understand the evolutionary relationship among animals. So let's begin with the first basis of classification, and that is the level of organization. See, level of organization refers to how cells are arranged in an animal's body to perform different functions. So based on the complexity, there are 5 levels of organization. First is protoplasmic level of organization. This is the simplest level of organization where the whole organism consists of only one cell. Since there is only one cell, all life activities like nutrition, respiration, excretion, reproduction, stimulation, all are carried out by that single cell. There are no tissue, no organs and no organ systems. For example, amoeba paramecium euglena. Okay, one question to you. If amoeba has only one cell, how does it digest the food? Yes, the answer is the digestion takes place inside the food vacuole within the same cell. So just imagine how one cell performs all the functions. Then the second level of organization is cellular level. At this stage, animals are multicellular but do not form true tissues. Instead, the cells are loosely arranged and work independently. The best example is phylum Porifera. So sponges contain different types of cells like conocytes, pinacocytes and amoebocytes, and each cell performs a specific function. For example, conocytes create water currents and capture the food, and amoebocytes transport nutrients. You know why sponges are considered as primitive? Because if a sponge's body is broken into individual cells, many of those cells can reorganize and form a new sponge. So this ability is called cellular totipotency. Now come to the third one, the tissue level of organization. As evolution progressed further, cells started grouping together and similar cells perform the same function, forming a tissue. So the phylum Cnidaria shows tissue level of organization. For example, hydra, jellyfish, sea anemone, etc. These animals possess epithelial tissue, muscle tissue and nerve tissues. However, organs are absent here. Now coming to the fourth level, that is the organ level of organization. So an organ is made up of 2 or more tissues working together for a specific function. For example, mouth, pharynx, intestine. So the animals belong to the phylum Platyhelminthes, show the organ level of organization. For example, planaria, liver fluke, tapeworm, etc. And the last complex level of organization is the organ system level of organization. Here, different organs work together to form an organ system. For example, digestive system, respiratory system, circulatory system, nervous system, excretory system, reproduction system. So this level begins in phylum Nematoda and contains up to Chordata. For example, Ascaris, earthworm, cockroach, fish, frog, human, etc. So now we have understood the level of organization Let's move to another very important basis of animal classification, and that is the body symmetry. See, if we draw an imaginary line through the body of an animal, can we divide it into two equal halves? Yes. The arrangement of the body parts with respect to an imaginary plane passing through the body is called body symmetry. Look at your own body. If I draw an imaginary line from the middle of your head to your feet, then your body is divided into two equal halves. But does every animal show the same arrangement? The answer will be no. Some animals cannot be divided into equal halves at all, while others can be divided into many equal halves. So there are four major types of body symmetry: asymmetry, radial symmetry, biradial symmetry, and bilateral symmetry. So animals with asymmetry is generally have an irregular body shape, so their body cannot be divided into two equal halves by any plane, because most of them are sessile, meaning they remain attached to a surface throughout their life. For example, Spongilla, Cycon in phylum Porifera. The second type of body symmetry is radial symmetry. In radial symmetry, any number of imaginary planes passing through the central axis divide the body into two equal halves. Just think a bicycle wheel. No matter where you pass a line through the center, both sides remain equal. Examples are hydra, jellyfish, sea anemone, adult echinoderms like starfish and sea urchin. But yes, another important thing, remember, the larval form of echinoderm is bilaterally symmetrical, not radial. Okay, so remember, this is a frequently asked question. Okay, then the next type of symmetry is biradial symmetry. It is modified form of a radial symmetry where the body can be divided into two equal halves only by two specific planes passing through the central axis, because certain body structures such as tentacles or complates are arranged in pairs. Okay, so for example, the phylum Ctenophora. You can see the picture present over here. The last type of symmetry is bilateral symmetry. This is the common type of symmetry seen from Platyhelminthes onwards, means up to mammals like humans. So here only one imaginary plane divide the body into two equal mirror image halves. So if we see the evolution, then the sequence will be asymmetry, radial symmetry, biradial symmetry, then bilateral symmetry. It shows a gradual increase in structural complexity and specialization. Now coming to the next characteristics classification, which is germ layers. Germ layers are the primary layers of cells formed during the gastrulation in early embryonic development. These layers differentiate into all the tissues and organs of the body. So there are three primary germ layers: ectoderm, mesoderm and endoderm. See how its formation occurs. After the fertilization, the zygote undergoes cleavage to form the zygote into morula, then blastula and finally gastrula. Gastrula is the stage where the germ layers are formed. The ectoderm is the outer germ layer, and the derivatives of ectoderm are nervous system like brain, spinal cord, peripheral nerves, etc. Then the sense organs like eyes, inner ear and nose, skin, hair, nails, sweat glands, sebaceous glands, enamel of teeth and the pituitary glands, etc. Then the middle germ layer is the mesoderm. It gives rise to skeletal system like bones, cartilage, muscular systems like skeletal muscles, smooth muscles, cardiac muscles, then the circulatory system like heart, blood, lymphatic vessels and excretory systems like kidneys, ureters, and the reproductive system like testes, ovaries and connective tissues, and the tissues like tendons, ligaments and the other like spleen and adrenal cortex, etc. Then the inner germ layer is endoderm. It mainly forms the lining of the digestive and respiratory system. For example, lining of stomach, small intestine, large intestine, liver and pancreas, etc. Then the respiratory system like trachea, bronchi and lungs. Then the endocrine glands like thyroid, parathyroid and thymus, and the urinary system like urinary bladder and urethra. All are the derivatives of endoderm. Now we will see the germ layers present in animals. See, diploblastic animals possess two germ layers, ectoderm and endoderm, and a non-cellular layer called the mesoglial. It lies between them. So examples are included hydra, jellyfish, sea anemone, corals, etc. All are belong to the phylum Porifera and Cnidaria. Then the triploblastic animals possess three germ layers, ectoderm, mesoderm and endoderm. Examples are included flatworms, earthworms, cockroach, fish, frog, birds, mammals, etc. It means all the phylum starting from Platyhelminthes phylum. Okay. Then coming to the next characteristic feature, and that is the body cavity, also known as coelom. So a coelom is a fluid filled cavity that is located between the body wall and the alimentary canal, which is completely lined by mesoderm. So what is the body cavity exactly? Just imagine a balloon. The outer wall is equivalent to the body wall and the inner tube is the gut, and the space between them is called the body cavity, means coelom. If that space becomes completely lined with mesoderm, then only it becomes a true coelom. So why the coelom is so important? It is important because it provides space to the internal organs that can develop freely. And the coelomic fluid also helps in transport nutrients, wastes and hormones. So there are three types of organisms according to their presence or absence of coelom. First one is the acoelomate. It means no body cavity. A means absence of coelom. So here body wall directly touch to the digestive tract. Examples are planaria, tapir, liver fluke, etc. All are coming under the phylum Platyhelminthes. And then the second category is pseudocoelomate. Pseudo means false. So false coelom, it means body cavity is present but not completely lined by mesoderm. So examples are roundworm, ascaris, Wuchereria, all are coming under the nematohelminthes. Then the last main category is eucoelomate. It means true coelom. Here body cavity completely lined by mesoderm. For examples, earthworm, cockroach, starfish, fish, frog, human, etc. It means onwards the phylum Annelida, and its advantages are highly developed organs, better circulation, better reproduction and complex organ systems. If we see the formation of true coelom, then it is of two types, schizocoelous coelom and enterocoelous coelom. Schizocoelous means mesoderm splits into two layers. This type of coelom seen in Annelida, Arthropoda and Mollusca. And the enterocoelous means outpocketing of God-formed coelom. So it occurs in the deuterostomes, for example, Echinodermata, Hemichordata and Chordata, etc. So this is all about the body coelom. If we see the evolutionary significance, then it progressed like acoelomate, pseudocoelomate and eucoelomate. Now coming to the last but not the least, the characteristics of segmentation or metamerism. It is the division of body into a series of repeated segments or metamers, arranged one behind another along the longitudinal axis of the body, like the couch present in the train. For example, the earthworm is the best demonstrate of true metamerism in Annelida. Here the body is divided into numerous segments. These segments are separated internally by the septa. So the metamerism segmentation can be classified as homonymous or heteronomous depending on how similar or different the segments are. In homonymous metamerism, homo means same or similar, meaning the successive segments are more or less similar in structure and function. For example, earthworm and leech. And in heteronomous metamerism, hetero means different, so it means the segments are different from one another. For example, all the arthropods like prawn, cockroach, etc. So this is all about today's class. We learned all the major characteristics of classification. I hope these concepts will make the classification of different phyla much easier for you. If you found this video helpful, then please like the video, share it with your friends and subscribe to my channel for more zoology lecture. Thank you for watching. See you in the next class.